Tēnā koutou, tēnā koutou, tēnā koutou katoa. Thank you so much for this opportunity to share our work that we've been doing with Far-UVC on Ag Research in New Zealand. I'm a scientist with the Food Systems Integrity team. Been working with Far-UVC and blue allergy light since 2019. Last year I presented some work for the blue LED and Far-UVC and how they work really well together to inactivate different viruses and different types of surfaces. I also showed that we could inactivate extended spectrum β-lactamase-producing E. coli in solution. These are critical on the World Health Organization pathogen priority list. And that the lights work synergistically to inactivate these Escherichia E. coli. I also showed that if you expose these E. coli to repetitive long doses of Far-UVC and dual light, that you could induce light tolerance. But we didn't induce light tolerance in the antibiotic sensitive controls that we used. I also showed that if you passage those cells numerous times, up to 5 times, that the light tolerance was continued to be exhibited in these Escherichia E. coli. So this showed a potential genetic mechanism for this light tolerance. And as we know, with traditional UVC at 254, the mechanism is the DNA damage. There's a lot of changes of GC-based pairs to AT, especially in genes that are known for DNA repair and the SOS response pathway. So we wanted to see whether or not Far-UVC was causing the same sort of damage to DNA and then these mechanisms for light tolerance. So before I did the genomic sequencing to look at the genome, I had to ensure that light tolerance was still being shown by these E. coli that had been stored at -80 for up to a year. So I took them out, resuscitated them, and then exposed them to a 30-minute more lethal dose of either dual light or Far-UVC, and compared them with wild type control. They were still showing some light tolerance, so they were harder to inactivate. If you passage them once or 5 times, that light tolerance continued to be shown. So at the same time as growing up these bacteria, I took some of that overnight culture and extracted DNA, did a library preparation, and then subjected them to short read Illumina technology sequencing or long read with Oxford nanopore technology with MinION. And then I was able to overlay those short reads with the long reads to get a complete genome and also placements that we could then do analysis with. And we used a number of different types of software to polish and ensure the quality of those sequences before analysis. So back in 2022, colleagues had already sequenced these 2 genomes, Ag Research 5151 and 6128. They had been isolated from freshwater sources in New Zealand. So their analysis is there on the left. The genome size is around about 5 million. There's 3 contigs, so there's the chromosomes and then 2 plasmids, a small plasmid and a megaplasmid. And then we did some further analysis to see what sort of differences there were between these 2 E. coli, and they're actually very similar. They only differ by about 2 gene clusters, so some transposases and some thermonuclease family proteins on the plasmid of 5151. So we could use these sequences to then compare our sequences once we've done our sequencing. So we have 6 for each of those 2 strains. So the original are shown in blue and green. We then had some controls that weren't subjected to the repetitive light but were passaged once or 5 times, which are shown in red and orange. And then we have the light tolerant to the dual light in purple, the light and dark purple, and then in black is just light tolerant to Far-UVC. So the genome and the chromosome sizes are fairly similar. There are some variations which is due to sequencing and the assembly. The important thing to note is the GC content is the same across all of them. So for traditional UVC at 254, the signature characteristic of that is a change from the GC to 18 base pairs. So we would have assumed to see that GC content change if there had been more changes in base pairs from GC to AT, but we didn't see that. So looking at the plasmids, this is the small plasmid. I didn't include the large plasmid because it's sort of very similar. There's not a lot of difference between the plasmids. You can see 1 to 3 base pair differences between each of those. The controls aren't really aligned with the original, so those changes aren't happening due to the light exposure, they're just happening due to natural mutations. And when we aligned the plasmids, all of the genes aligned fairly closely. You can see there's these CPG islands, and there's a lot of those on the plasmids. And these are important as sites for DNA methylation, and different patterns of DNA methylation then cause differences in gene expression and protein expression. So we're thinking that there could be some differences in that DNA methylation due to light tolerance, but that's something that we need to have a look closer at to see whether it's involved in this light tolerance or not. So there have been some light-resistant genes that have been published previously for traditional 254, REK, XAUS, so some of them are involved in that DNA recombination repair as well as the SOS stress response. So we had a look at all of these genes in our light-tolerant strains and found that there were no single nucleotide polymorphisms or SNPs involved in any of those genes that have been previously identified for that traditional UVC light resistance. And further gene expression studies are going to be needed to see whether or not any of those genes are upregulated after that exposure to the repetitive pulses. So some of the point mutations that we did find, there weren't a lot, and those that have similar point mutations across all of the strains are not just occurring in the light tolerant, but also occurring in the controls, so they're not likely to be due to the exposure to light. There's quite a few intergenetic changes happening, so those are regions in between genes. These are areas where you might have promoters or protein binding sites or splicing sites. So they could be important in terms of changing the gene expression and protein expression. Again, these are areas that we need to have a closer look into to see if they are of importance. So why do we want to learn about light tolerance? Why is it important? It was already raised earlier this morning about whether or not bacteria can become light resistant. So I did some whole rooms with disinfection studies where I was given a my lumens unit that I placed on the wall, 1.3 metres and down onto the bench. I took those two bacteria and plate dropped them onto SBA, and they had about 50 colony forming units per drop, and I exposed them to 2, 4 and 8 hours of this Far-UVC light that cycles on and off, so I had a 30-second on, 50-second off, and it continued to do that if you waved at it. So it was a long day for me and waving at this light. And what we saw was that over 8 hours the E. coli did reduce, but was still present after that 8 hours. Now SBA is quite a nutrient-rich agar, but I wanted to hopefully see some survivors so I could then do some further experiments to see if they were going to show any light tolerance. So I took those colonies and the controls, which should be on the other side of the agar plates, and injected them to a more lethal dose of Far-UVC, and saw that there was no light tolerance that had developed over that 8 hour period from those survivors. It's not to say that light tolerance can't be induced if those bacteria are continually exposed for longer periods of time, but within that 8 hours I didn't see any difference between those and the wild type that were treated. So in conclusion, light tolerance wasn't impacted after prolonged storage at -80 to 12 months. Using long and short read sequencing methods, we could see minor differences between the light tolerant strains and the controls. There were some point mutations, but the significance of those genes are not known yet. We have to look into that further, as well as those intergenic regions. Further epigenetic analysis of the light tolerant and the controls is needed to determine whether there's any impact of DNA methylation on gene expression. There were no point mutations found in any of those putative light-resistant genes that had previously been published for traditional UVC resistance. Mutations in the genome are likely not the cause for the light tolerance, but further work needs to be done on this. Reduced survival of the EC strains was seen after that 8 hours of whole horrendous infection, but was still present, and that no light tolerance was observed after being exposed to that cyclic on and off Far-UVC for 8 hours. So some future work, want to investigate the genes harbouring the point mutations in those light-resistant strains. I can use CRISPR-Cas9 system to induce some targeted mutations into those genes or the intergenic regions and see what sort of phenotype they show. Could use RNA-seq to assess gene expression of light-tolerant strains in comparison to the controls, and also look into that DNA methylation and its role in light tolerance. I just wanted to acknowledge the co-authors and Rose Collis for helping me with my sequencing, and also to the organising committee and sponsors, and also the NPOR for providing some funds to get here. Thank you.